So this is our lecture on the DNA's structure and how it is able to be replicated. So to begin, we need to talk about nucleic acids. Remember from biology in ninth grade that nucleic acids are one of the four macromolecules. The nucleic acids that we find in living things are two different forms. There is DNA and there is RNA. DNA is found within the nucleus of every cell of every living thing, and it is going to contain the primary carrier of genetic information. So, in other words, your DNA is sort of like your original recipe book. It contains all of the information that is needed in order to make every protein of every trait that builds you. RNA is slightly different from it, though closely related, also a nucleic acid molecule, but there are different forms of RNA and they do different jobs. For example, messenger RNA is going to play a role during transcription. You have ribosomal RNA that's going to assist in protein synthesis, and transfer RNA that will also assist in protein synthesis. Also, recently we have discovered that there are small interface RNA molecules that are going to be able to do a variety of different jobs. So let's take a look more closely at what it means to be built of nucleic acid. That means we're going to look at the monomer of nucleic acids, which is a nucleotide. So all DNA and RNA molecules are made up of single subunits called nucleotides, and this is the basic shape of a nucleotide. It contains three parts. You have your nitrogen base, you have your phosphate, and you have a sugar group. So all three of these are found within a monomer. So let's take a closer look. In the sugar molecule, we see one difference between DNA and RNA, and that's because in DNA, we're going to use a sugar known as deoxyribose, whereas RNA just uses ribose. And if you look at their structures, the only thing that you can see that is different is the fact that coming off of the third carbon, and when I'm counting, I'm saying this is carbon 1, carbon 2, and carbon 3, we have an OH group, just like we do in ribose, but coming back off of the second carbon, notice that we're missing an oxygen. So that's where we get the name deoxyribose from. All monomers also have a phosphate group. While the phosphate group does not change between monomers, it is always the same. It does play a very important role, especially in biotechnology, because our phosphate group is what's going to give DNA its slight negative charge. And this will be important in a technique that we're going to be learning next week called gel electrophoresis. So if we look at this chain here, hopefully what you can see is that it is made up of four monomer subunits. So this is a polymer of a single strand of DNA, and it contains four different nucleotides. So we have the nitrogen base, sugar, and phosphate group. That's one. Here we have another nitrogen base, sugar, phosphate group. Here's two, right? Then we got three, and then we have four. Okay, so we see four nucleotides in this picture. But so far, the sugar and the phosphate really are not what make the nucleotides diverse. It is the nitrogen base. So there are 5 different nitrogen bases, and they have 2 different structures. So take a pause here, make sure you get all of this information copied down, and then let's go look at the next page together. So here are the 5 nitrogen bases, and they are drawn out to show the structural shape of them. On the top, you see we have what are called purines, and that would be your adenine and your guanine. And then on the bottom, we have what are called pyrimidines, cytosine, uracil, and thymine. There's a distinct difference between the purines and the pyrimidines. Can you figure out what it is? It's the fact that the purines have what we call a double ring structure, and the pyrimidines have a single ring structure. This will become important when we start taking measurements on the DNA molecule, which we will talk about in just a few slides. Also, it's important to notice that when we look at DNA, DNA is made up of A, C, G, and T, but RNA is made up of A, U, C, and G. So they have A, G, and C in common between DNA and RNA. But then instead of thymine, we see uracil in RNA. And if you look at their differences structurally, uracil and thymine, they're not really that different, right? A lot of the same things. The main difference there is the fact that coming off of your fifth carbon, you have a methyl group in thymine, whereas in uracil, it's just a hydrogen. We don't know why there are these differences. Sometimes the reasoning is more discovery based, and what that means is we just make observations and we see that they're consistently applied. In every DNA molecule, we see the use of these thymine structures rather than uracil. So let's start talking a little bit about history. The race for the double helix. So hopefully by now or tomorrow, I'm not sure when, you will have done the timeline activity where you will learn about how long we've been looking into what this magical material is inside of our cells that determines our traits. By the 1950s, we had figured out that it was the DNA found within the nucleus, and we knew what that material was. But now the race became to determine the molecular structure. So, who are the main players involved? Watson and Crick are probably the names you're most familiar with. Then next, we also have Rosalind Franklin and Erwin Chargaff. All four of these individuals contributed to the discovery of the structure of DNA. However, Watson and Crick are the ones who won the Nobel Prize alone. So let's jump into this story. So first we have Erwin Chargaff. He took a bunch of different organisms, he extracted their DNA, and then he analyzed that DNA chemically. And what he looked for was how many of each base was found in the DNA molecule. So pause the video here for a second and just analyze that data. Pretend you're Chargaff. What trends or patterns do you see in the data? So coming back from that pause, what you should see is that for the majority of the organisms on this chart, the amount of A, like the percentage of base A, is very close to or equal to T. And the percent of G is very close to the percent of C. So this is actually a really important thing, and we'll talk about it in a few slides again. But unfortunately, when Chargaff discovered this, his research was found to be of no significance because people said, yeah, okay, so what? Who cares if the amount of A equals the amount of T? He didn't have a chance to put together the meaning behind the evidence. Rosalind Franklin was a female scientist in the 1950s. She worked in a lab underneath another gentleman, Maurice Wilkins. I always think it's important to point out that she actually had more experience and more credentials than Maurice Wilkins, but because she was a woman, she was not allowed to run her own lab. Anyway, Rosalind was very interested in doing X-ray diffraction of the material of DNA because she wanted to see what the actual positioning of the atoms within the molecule could tell us. And here on the bottom right is her famous photo 51. And what we're seeing is a pattern. And so we know that when we take an X-ray, right, what happens is when the X-rays are able to go through, they're going to leave a black shadow. But when they hit something solid, like our bones, then they're going to show white. So she used a special kind of X-ray. She used crystallography, and she was able to make this pattern. And so what it shows is you could almost draw it here if this X pattern makes a spiral shape, okay? And she took all sorts of measurements on here to determine where the turns were within the spiral and how far apart they were and how wide the molecule was. And she came to the conclusion that the molecule was helical shaped, okay? So it looked like a spring. James Watson and Francis Crick were also working on the structure, and there's a little bit of drama involved here because James Watson was good friends with Maurice Wilkins, and the story goes that he came to pick up Maurice Wilkins for lunch one day, and he walked into the lab where Rosalind was doing her work, and he looked at her desk and looked at the photo. In fact, here is an excerpt. Just take a second to read that. He admits that he looked at that picture, and that by looking at that picture, he was able to determine, yes, indeed, that the molecule was helical shaped. So he looked at her work before she had even published it. So there's a lot of controversy surrounding that. Without looking at that photo, would he have been able to figure out the structure of DNA? Nobody knows. But Rosalind Franklin sure was angry about it. Anyway, within a month, Watson and Crick, they were the first to publish the paper. They got all of the fame and the fortune and the credit and the Nobel Prize for discovering the structure of DNA. So how did they do it? Well, work smarter, not harder, by looking at Franklin's data and then Chargaff's published data as well. So the X-ray photo supported the fact that the width of the DNA molecule was uniform. So how could we get base pairing between the nitrogen bases and still have the width of the DNA molecule be uniform? Well, if you're always pairing a pyrimidine, which is a single ring, with a purine, which is a double ring, then it's always going to be 3 rings wide. Put that together with Chargaff's data, he noticed that A and T were equal and C and G were equal. But he came to the wrong conclusion. It wasn't that they were equal, but it was that they always paired together. So anytime you found an A, you also had a T. And this makes sense. Now, if you look back at Chargaff's data, some of his numbers weren't exact, and that's because in science, numbers are never 100% nice, round, whole numbers. The differences are there because of mutation. There are instances where sometimes there's a mistake in our DNA, and instead of properly pairing A and T together, maybe A and C get paired together. And so that happens every so often, and if it doesn't affect the survival of the organism, then that mutation will be passed on. So let's take a look at this. Here's an example of a math problem. As long as I give you the percent of one of the 4 nucleotides, you should be able to figure out the percentage of the other 3. So here, if I told you the amount of adenine nucleotides in a piece of DNA equals 40%, you should be able to figure out the other 3. So right away, we know that A always pairs with T, therefore their percentages should be the same. So that's 40 and 40. But we know nothing about G and C except that they should equal one another. However, there's a number here we're not realizing. A, T, C, and G percentages all have to add up to the whole DNA molecule that you're looking at, which means the total has to be 100%. So already, if A and T make up 80% of the molecule, then that means G and C would make up the remaining 20% of the molecule. And then since G percentages are supposed to equal C, you would take that 20% and just divide it in half equally. So we know that both G and C are 10%. So the shape of the DNA molecule here, we're looking at a double helix, or what we call a twisted ladder. Here's an image. This is a really good diagram. Put a star next to this one and make sure you study it a little bit. We can see a nucleotide here. We see a phosphate group attached to the sugar group attached to a nitrogen base. And in this case, the nitrogen base is a single ring structure. And this one happens to be thymine. Attached across the way to it, base paired together, is an adenine nucleotide. So what holds the 2 strands of DNA together? It's simple but weak hydrogen bonds in the middle. However, when we're talking about a DNA molecule that's 1 billion base pairs long, we now have, 2 billion, well, actually, it's more like 2 to 3 billion hydrogen bonds within the same molecule. So even though they are considered weak bonds because of the sheer number of them, it's actually strong enough to hold the structure of DNA together. So the phosphate sugar zigzag shape here makes like the backbone, or sort of the sides of a ladder that run vertical, and then going across the middle are your base pairs, and the base pairs are held together with hydrogen bonds. So we showed you a picture of the DNA molecule before on the previous slide, and it had these weird numbers, 5 and 3. What do those numbers mean? That's kind of weird, right? Pause the video here and just take a look at this diagram and tell me how the top strand of DNA differs from the bottom strand. What pattern do you see? All right, coming back from that pause, what we are going to see here is that when we look at this nucleotide, let's look at this first one here. We see the phosphate group hanging off of the left. We see the sugar, which is sort of a pentagon shape here, with the oxygen also hanging off of the left. But when I go on the bottom here, I don't have my phosphate on the left. My phosphate is on the right. And when I look at my sugar, the oxygen is also pointing to the right. And the left and right are relative just to this image. But what you can see is that the way that the nucleotide is oriented on the top is a complete opposite, the mirror image of how, not mirror, is flipped from how it is laid out on the bottom. So if you hold your hands up sort of so that your palms are facing each other but slightly apart, what you want to do is you want to take and turn one hand upside down. Okay? So why is this occurring? Why do we have one strand of the DNA having its nucleotides pointing in the other direction compared to the other side of the DNA? Then it has to do with those hydrogen bonds. In order to get these hydrogen bonds to match up here, this nucleotide has to be flipped upside down so that this bond can form. Okay? So now the nucleotides are pointing in this direction on this side and are pointing in the opposite direction on the other side. Same thing here, except we've just turned the molecule picture on its side. Okay, so now we're looking left and right, and notice that over here we have these weird numbers 5 and 3, but then down here we have 3 and 5. So let's learn a little bit more about this. So now we know why the nitrogen bases are pointing in the opposite direction. Now let's investigate the how. Those numbers, what they represent is, if you're looking at a nucleotide that is in the middle of, if you're looking at a nucleotide, in the middle of it we have our sugar group, right? And then coming off of it we have a base, which I've just reduced to the word base here, and the phosphate group as well. The nucleotide contains a sugar with 5 carbons. And so we start the numbering the carbons in a clockwise direction. And when you do that, you start with number 1, which is attached to the base, down to 2 here, 3 here, 4 here, and 5 here, because there's a little methyl group that attaches to your phosphate group up above. So why are 5 and 3 important? Well, when you look at this orientation here, okay, this is the 5' carbon, and this down here is the 3' carbon. It's kind of a little north and south, right? So let's go back to this picture, right? It looks like this one is in the opposite place from this one. And so scientists looked at this and they said, well, that would help us determine the direction, right? We could say this is the 5' end, and this down here is the 3' end. Okay, so that's how we determine the 5' and 3' end. So we're on one side of the molecule, and we've got our oxygen up here, right? And so we would sort of say, if we're running from 5 to 3, then that moves in this direction down the page based on how we have our image. But let's look on the other side of the DNA molecule. We're upside down, right? Because remember, we flipped that nucleotide. So we're still going to number using the same pattern, starting at the base, carbon 1, around to 2, 3, 4, and then 5 is attached to the phosphate group, which is now on the bottom. And what do we see? If we're looking at 5 to 3, it's now going in the upwards direction. Okay, so blowing this up, overall, here's a nucleotide. Where the phosphate is its 5' end, and where it attaches down here is its 3' end. So 5 to 3, 5 to 3, 5 to 3. So overall, 5 to 3 on this side. And then down here, the same thing, but just going in the opposite direction. So we have 5 to 3. Why does any of this matter? The most important thing, big star up here next to this bullet point right here. This anti-parallel orientation matters, and we track it very carefully because when building strands of nucleic acids, so when building DNA or when building RNA, they can only be built in one direction. We can only add nucleotides onto the 3' end of another nucleotide. Okay? So imagine that, I'm trying to think of something. Imagine you're snapping beads together, all right? And kind of like Legos. No, maybe like Legos. I'm not sure. But you can only add on to the 3' end. So what that means is here is the 5' end, and here is the 3' end. I can't bring a new nucleotide in here and attach it at this point, okay? This part of the phosphate group is inactive. I can only add here, all right? So I could add more nucleotides coming off of this 3' end. But down here, this isn't my 3' end, this is my 5' end, so I can't add on here. So instead, I'm going to add on down here at this end. Well, that's crazy because what it means is I can extend on this strand going in the left direction, but on this strand, I can only extend in the right direction. So this is going to be a very big problem when we start talking about replication. So replication is how we take a single DNA molecule and produce 2 identical DNA molecules from it. It's really important to understand we're not copying. We are instead taking the original molecule, we are splitting it in half, right? Here you see our blue strands represent the original double helix. We're going to rip that helix in half, and then the yellow represents a new piece of DNA. So this is a newly synthesized strand, and each original strand is going to be able to pair up with a newly synthesized strand. So note that because the blue pieces are being used in our 2 new DNA molecules, the original DNA molecule no longer exists. So it's not that one molecule copies and we end up with 3 molecules total. No. One original molecule splits and new pieces are built in order to end up with 2 new molecules, both which are identical to each other. So how does this happen? Well, we've got 3 key enzymes that are going to play a role. DNA helicase is going to come along and break the hydrogen bonds that hold the 2 strands of DNA together. That's going to allow them to separate, and we're going to hold them apart so they sort of make like a Y shape. Once they're held apart, those 2 strands are separate now, okay? So here's one strand, right? And these are all of the bases on that single strand. but those bases are now free to form new hydrogen bonds. So this enzyme here is called DNA polymerase. DNA polymerase is going to pull free nucleotides from within the nucleus that are just sort of floating around, free hanging there, and it's going to attach them to build a complementary strand to the original strand, okay? And then lastly, we're going to have an enzyme called ligase, which is going to pull all of the pieces together. So when looking at this slide, the blue represents our original piece of DNA, right? We have two strands that are originally together. Then helicase has split them apart, okay? So we see them separate. The DNA polymerase molecules are green, and there' one on each side here. And what's happening is DNA polymerase is adding the new nucleotides. But remember about the 5' and the 3' that we talked about? They can only be added onto the 3' end. So on this side, we're moving in the right direction, adding nucleotides. See the arrow pointing this way? Adding nucleotides in this direction, which is the 3' end. So everything's nice and smooth on this side, and we call this the leading strand that is being synthesized. However, over here, look where your 3' end is. It's at the opposite end. And so what that means is if helicase is moving in this direction, right? It is breaking those hydrogen bonds here and moving in this direction, sort of up. But this polymerase is moving in the opposite direction. What's going to happen? It's going to run out of space for where it can synthesize new strands. So what happens on this side, which is called the lagging strand, is that it's synthesized in these short fragmented pieces. And those short fragmented pieces then need to be sewn together by this enzyme DNA ligase, so that in the end, you don't see these individual pieces, but instead it will look like the leading strand. So, just to review, we take the original strand, here it's in red, we're going to split it apart using DNA helicase. Then DNA polymerase is going to add new nucleotides to a newly synthesized strand, right? And this is happening twice because we've got two molecules being made. Those newly synthesized strands are complementary to the original. And so our end result is not one, but two DNA molecules that are identical to each other. How do they compare to the original? Well, each new molecule, the whole molecule here, is made up of one original strand and one newly synthesized strand. We call this the semi-conservative model of DNA replication. And the reason we call it this, semi meaning half, conservative meaning save. So we are saving half of the original molecule, right here. Here's half and here's half. We're saving half of the original molecule in each new DNA molecule. So let's take a look at this in action, because it's very difficult to talk about these parts that are moving when we're looking at static images. Oops, sorry. All right, so what you're seeing here is a molecular model animation. Just take a second to look at it, and let's pause it here. Oops. Nope. Nope. Okay, we're gonna watch it again. Look at all these different little pieces color-coded, and stop. Okay, so let's take a look at what we have going on here. So first of all, I see double-stranded DNA coming in from this side, and this blue guy here is spinning super fast. And when the DNA emerges from the other side of it, it's a lot thinner. So to me, the DNA is being split by that blue spinny thing. So that would be your helicase molecule. Okay, this is annoying me. Hold on one second. I'm going to resize this so that I can access my pause button. Alrighty. There we go. All right. So here's another angle. Let's get rid of the ads. Here's where that blue is spinning, right? The DNA is coming in here and it's being separated. These green and purple, these are your polymerase molecules. And see if you can tell the difference between the leading and the lagging strand. Okay, so over here we see DNA molecule coming in, and then we've got it leaving in two different directions. Oh, hold on one second. Down here, this one is being synthesized continuously, whereas up here, look at this weird loopy thing. And can you see how it's growing in the reverse direction? So the one that's looping out, that's your lagging strand. And the other one is your leading strand. So this big blue enzyme is helicase. It's spinning and breaking those hydrogen bonds to separate the DNA molecule. Then coming off at the bottom of the movie, that's your leading strand. Notice that polymerase is easily attaching new nucleotides, and what we're producing coming out of here is a smooth, continuous piece of DNA. Okay, but the other half is the lagging strand, and you see this weird looping thing where you get the polymerase molecules jumping off and on again, right? Notice how that loop is going out. That's because the polymerase molecule is working in the opposite direction of helicase, and it's producing these fragments, these Okazaki fragments, and those fragments are then eventually going to have to be sealed together. I'm going to post a link to this video so that you can watch it also with the sound on, so you can get a little bit more description from this, but I just wanted to go over that quickly. Pause your video here and move to the next slide and see if you can fill in the answers. When you're ready, hit play again and the answers will be revealed. All right, here are all of the answers to the review quiz. You should have been able to find each of these. If you're having any trouble with this topic, please let me know. Remember, I'm available for office hours Thursday and Friday, noon to 1. All right, that's the end.